Hi, everyone. Welcome back. So, there's a lot of different reasons why we might want to grow bacteria in a laboratory setting, like we're going to do in our class. The first is the one that we're going to be dabbling with a lot here, which is identifying a microbial organism to species. Super important if we want to, say, treat a person who has a specific infection. That specific infection has a specific cause. And if you identify the cause, down to species especially, you can really tailor an effective treatment to help them. But there's a lot of other reasons too. A lot of researchers out there are actively studying bacterial form and function, their behavior, their ecology. They're doing genetics research through collecting their DNA and analyzing it. They're studying the evolution of bacteria and other forms of life, and also looking at their metabolic activity, looking at what they can make, what they can't, what they can digest, what they can't, and of course their relationship to disease as causative agents. But there's also really important reasons to keep living collections even when not for study. For example, the place that we get all of our insulin nowadays is actually from E. coli collections. Humans produce insulin, but there are some people, diabetics, that can't produce insulin or can't produce enough of it and need to inject insulin to stay healthy. And it's not something that can be easily acquired. It used to be that to collect insulin and for medical use, people would have to harvest it from like pigs. So you would collect pancreases from butcher shops and things like that. They had a relationships with these companies that would essentially allow them to collect these materials and extract insulin from them. It was a whole bunch of issues around that. It wasn't very cost effective, most of all. And so insulin was a hard resource to come by and it was very expensive. But we later figured out how to take the gene for human insulin and insert it into the genome of an E. coli strain. And now we can grow the E. coli, in copious quantities in a laboratory setting, industrially, and then extract the insulin from them at a later time, which is frankly amazing. So there's a variety of reasons why we would want to do this. To accomplish this goal, we have to grow bacteria on special culture media. Okay, this is nutrient-rich substrate of some kind that is used to grow microbes specifically. And a lot of prokaryotes that are out there, versus this huge diversity of microbial life, have never been successfully cultured, never been successfully grown on this culture medium. And there's a few different reasons for that. I mean, the first one is that a lot of species have simply not been identified by science. And so it's hard to grow something that you've never observed. Not impossible, of course, but it's hard to do that. But the other issue is that microorganisms can be very picky. A lot of species require very specific things from their environment in order to survive and to reproduce. They need specific food resources, specific nutrients that they can digest. As I mentioned earlier, not all resources are equally accessible. There are some things that some species will not be able to digest, but others will. They need a specific amount of water. They need a lack of certain toxic substances or detrimental environmental conditions, things that will result in their death if they're exposed to them. And that can also vary from species to species. What is toxic? What is detrimental? Can be incredibly variable. They need the right temperature, the right pH, the right atmospheric pressure, things like that. Sometimes they even need other species to be around, specific partners or specific prey species or specific hosts and so on. So there's an incredible variety of these microbial needs, what they need from their environment. And that isn't always very easy to capture inside of a growth medium for us to grow bacteria in. We have a variety of different culture media to accomplish a bunch of different functions. Some of that is to help specific things grow under those conditions. Some of that is to help us differentiate qualities of things that do grow on these media. So they serve a bunch of different functions for us in our in our purposes of growing these species in the lab. For our purposes, we're going to use a bunch of different kinds of media, but they mainly come into like two categories. We have gel media and we have liquid broth. So gel media is made with agarose. This is agarose is a plant polysaccharide. It's a sugar chain that can come together and when it's dissolved in a liquid solution, and especially after it's brought to a boil, it can turn into a gel surface, so that it's like semi-solid. It's not rock solid, but it's squishy and capable of deformation, and it's something that bacteria could grow on or swim through in some cases. So it's a pretty cool medium. This is very similar to the kind of stuff that you would get in like a vegetarian Jello or something like that. So it's pretty safe actually, agarose. And then we could also have these same media in liquid broth form, as in it doesn't have the agarose. There's no agar in it. So it's just a raw nutrient broth with whatever specific additions it needs to accomplish whatever specific goals we have for that media. So this lecture is all about these types of media: the general purpose, selective, differential, and selective and differential media that we're going to go into here. So the first one we're going to talk about is the general purpose media. Okay, so this is culture media that is suitable for the generalist, the non-picky microorganisms. We call them non-fastidious sometimes. So these are the microorganisms that we're maybe most familiar with because they like the kind of conditions that are easy to come by. So this general purpose media, this is the gist of what it really contains. It contains a source of energy, carbon. Typically that's glucose or another sugar. It contains assorted amino acids and vitamins that are important for the bacterial growth. It contains some water, particularly in the liquid broth, but also dissolved in the gel, or like inside, as part of the gel. And then they also have a small amount of salt. Too much salt can be problematic. Too little salt can be problematic. All life needs a little salt, and so there's a little salt in there. And so the example that we're going to be most familiar with is tryptic soy agar, okay, otherwise known as TSA. We see this in a couple of our labs. We're going to see this pretty often, but this is a very common general purpose media that will grow a lot of stuff. It won't grow everything, as we talked about before. There are some things that need conditions and resources that are not in TSA, and we won't be able to grow them. Everything that does grow on a TSA plate or in a TSB broth, which is the same thing but without agarose, all of that stuff that grows there is a generalist. It is non-specific. It can handle diverse conditions and is pretty non-picky. For fungi, we have another kind called potato dextrose agar that's pretty common as well. Okay, but we won't really mess with that one. This general purpose media, okay, again, is really just for growing generalist microbes. But it's also a really useful way of determining the colony morphology of the things that grow there, as in, what's the shape of the colonies that form? What's the color of them? What is the opacity? Like, are they see-through or not? Do they have any unusual texture to them? What's their size? Things like that. Those characteristics can be diagnostic. It can help us differentiate one species from another. So we can employ these methods to like spread bacteria really thin on these plates and help them form colonies that are very isolated from one another and that can help us assess those characteristics. Now, because bacteria reproduce asexually, as in they don't have a partner when they reproduce, they just copy themselves, we only really need a single cell to create a colony. Okay? Only one cell is needed to be on that medium in order to initiate growth, just a single cell. And ideally, we only have a colony made from or starting from a single cell, because then it will grow in a very uniform way that allows us to assess this colony morphology. Now, to isolate these cells and to do this process of isolation for the purposes of colony morphology assessment, we have to do this thing that we call streak plating. Basically, what we're doing is we take a loop of our bacterium, the specific species that we want to grow, and we streak it across a quarter of the plate, and then we sterilize that tool. We put it over an open flame. We kill all the bacteria on that loop. Now, we don't go to our original sample that we used. Everything we need is now on this plate that we're working on. What our goal is we're going to take what's on the plate now and we're going to spread it out further so that it becomes, the bacteria are less and less concentrated with each additional quarter of this plate that we spread the bacteria out on. And in the end, what we hope to see after the bacteria grows is this. In the section that we first apply the bacteria, growth is going to be very dense. There will not be any distinct colonies that form. If we keep on moving in the direction that we spread out the bacteria, it'll be a little bit more, a little bit less dense, a little bit more spread out, even more, and then eventually at the very end, at the very furthest and most diluted, we spread out the bacteria in our original streak plating. We should get just a handful of isolated colonies that we can use to actually help us assess that proper morphology. That can be very important. Now, aside from general purpose media, we also have selective media. And as the name implies, this is selective. Its job is to inhibit the growth of certain microbes but not others. It literally selects for specific things to grow. This is because of the property of that specific medium. So there's going to be things inside the media or qualities of it that are going to be ideal for some species and non-ideal for others. The ones where it's not ideal for, they're not going to grow. So we could be looking at something that, the media might be high or low pH. It might have certain antibiotics or antiseptics or sterilizing agents in it. It might have specific salts or high quantity of those salts. It might have other resources that are really difficult to metabolize, but some species can do it and others cannot. And a variety of other things that might be in here that could help deter the growth of some things but promote the growth of others. So we're going to use some different kinds of selective media in our class. We're also going to look at differential media. Okay? Differential media differentiates between the organisms that grow on this plate. So there's nothing selective going on here. This is still a general purpose media in the sense that everything can grow here as long as it, the things that are non-fastidious, the things that are not picky, can still grow here. But there's a quality of the plate that creates a difference between some species and others that is observable to us. So a good example of this would be the blood agar plates. So this is TSA, it's a TSA plate, plus a 5% concentration of sheep's blood. Okay, so it contains red blood cells that are inside this plate. It gives the plate a red color. This is what blood agar is. Sometimes this is even called TSA-B. TSA-B, because it's TSA with blood. Now, some species of bacteria can produce these exotoxins that we call hemolysins. These are special molecules whose job it is to destroy red blood cells. So We see these in species that are pathogenic or opportunistic in some cases. So these hemolysins, they vary a lot in their form and function. There's lots of different kinds, and also their effectiveness, like how effective they are at destroying red blood cells. Some can damage the red blood cells, like poke holes in them and hurt them, but not destroy them outright. And then some can completely break down and destroy red blood cells. And then some species won't make hemolysins at all. So there's a lot of variation here. So when we look at this as a differential medium, the bacteria can be either incomplete, they can do incomplete lysis, where they break down the red blood cells, but not all the way. They could completely lyse the red blood cells, where they are destroyed and it results in the clearing of the media. Or they could not do any form of lysis, where like they don't have hemolysins, they don't break down any red blood cells. We call this, in this order, alpha, beta, gamma. Okay? Now, this is important and easily confusable. All right, so make sure to practice this. That alpha is incomplete breakdown, beta is complete breakdown, and then gamma is no breakdown at all. Now, the way we assess this over here is we look for how the media reacts to the growth of this bacteria. So the bacteria, if they have hemolysins, they will secrete them into the environment, onto the plate, and that will have specific results. So alpha hemolysis, because it's incomplete, will incompletely break down the red blood cells. It'll cause the gel, the medium, to turn a green or brown color, and it'll become a little bit lighter usually. Beta hemolysis is a bit more pronounced. We have the clearing of the media. Again, all this stuff, it happens in a halo around where the bacteria is growing. So you'll actually be able to see the regular TSA in the halo through this, where the bacteria has done a lot of breakdown of these red blood cells. And then for gamma, you won't see any change at all. It'll look exactly the same as any other place in this plate where bacteria does not grow. Okay? So this is why we call this a differential mediums, because all these species, whether you're alpha, beta, or gamma, they can all grow here, but the result is different, as in like they have a different impact on the media. Another important media type that we want to look at, and one that we're going to be spending more time with, is the selective and differential media. It is selective and differential, so it has qualities of both. So there's one aspect to a selective and differential plate that is selective, meaning that it prevents certain things from growing and allows others to grow. And then there is another aspect that is differential. Of the things that do grow, what is the difference between them? Right? That's essentially what's happening here, what we're going to be evaluating. So there's two important ones that we want to talk about. The first one here is the mannitol salt agar. And we will be using these, they'll be very important for us, and we want to make sure that we understand what these plates do. So the mannitol salt agar, or MSA, is a plate that has a high salt concentration. It's very salty medium. It has like 7.5% salt concentration in it, which is very high. Only species that are halophilic, as in they love salty conditions and they live mainly in salt or are very tolerant of salt, are able to grow there. Things that cannot handle high salt, they're not going to grow. Now, these halophiles that do grow, the mannitol salt agar plate is also going to differentiate them by whether they can ferment mannitol. So we have a sugar that is inside of this plate. The sugar is mannitol. Okay. Now, if the plate If the bacteria that grows here can digest and ferment mannitol, it will release an acid byproduct that will react with a pH detector, like an acid detector that is in this plate, and as a result, it will turn yellow. So in this example, we have two species here, Staphylococcus epidermidis and Staphylococcus aureus. Now, epidermidis, ignore this is spelled incorrectly, it's epidermidis. So Staphylococcus epidermidis cannot ferment mannitol, and so when it grows on this plate, it does grow because it loves salt. It grows on the surface of our skin, which is, we are prone to sweat and we release salt onto the surface of our skin. Our skin is a salty environment. So they grow on our skin, but they can't digest mannitol, and so there's no breakdown here. There's no acid production from the fermentation process, and so there's no color change. But Staphylococcus aureus also still loves salt. It grows on our skin very normally. But this species does have the ability to digest mannitol and ferment it. And so it does this. It releases an acidic byproduct, which reacts with the pH detectors in this medium and turns it yellow. Okay? So that's the main difference between these two species and why we can differentiate between species that are able to grow on this environment. We have another selective and differential medium that we want to talk about, and that is the EMB plate, or eosin methylene blue. Okay? Now, this is a type of medium that's pretty cool. It is selective for gram-negative species. This methylene blue in particular, but also the eosin to some degree. These are chemicals that we've added to the plate that have an antibiotic quality to them, particularly targeting gram-positive species. And so they can prevent the growth of gram-positives while allowing gram-negatives to grow. So now that's a really important thing that we can do with this, that we can actually, right off the bat, we can separate gram-positives from gram-negatives without even necessarily having to gram stain anything. Then there's a differential quality as well. We can differentiate bacteria that do grow on this medium by their ability to ferment lactose. Okay? And so there's actually some variety here. If the bacteria can ferment lactose rapidly, there's going to be a lot of fermentation byproduct production, which is very acidic. And so it can react again with these dyes that have something of a pH detection, and the result is that the colonies of bacteria, if they are doing this rapid fermentation, they can dye they can be dyed a metallic green color, and they get very shimmery, and it's quite pretty. Species like E. coli is capable of this, so if we have an opportunity to look at this in class, that can be very indicative. But there's a lot of species that can also do this process. It's not just E. coli. That's all that it means, is if you get this color change, you get this shimmery pattern that emerges. All it means is the bacteria is very rapid in its ability to ferment lactose. Then, if the bacteria cannot ferment lactose at all, the colonies are going to appear in a very colorless formation where, like, they're here, they grew on the medium, but they're transparent. And so you can see right through them, and there's nothing, there's no color change to them. That's indicative of them not being able to ferment lactose at all. Some species can ferment lactose, but they can't do it very fast. So that in those situations, you can exhibit color changes, but they won't necessarily be, this like beautiful green, metallic green color. They can be other combinations of colors. So in medicine, we can sometimes use this medium to help differentiate between coliform bacteria. This is a group of gram-negative species that's pretty commonly associated with fecal matter. It's not they're only places they live, but that's a common association. So they're intestinal bacteria. Some of them are pathogenic, some of them are opportunistic, and some are simply just normal flora that never cause any problems. This is an example here of two different species. So Proteus mirabilis over here is lactose non-fermenting. And so when you grow it on an MSA plate, an EMB plate, you grow it on an EMB plate, it grows because it's gram negative, but there's no color change associated with it. But E. coli, that's a common species that occurs in our gut, and it when it grows, it's gram negative. When it grows, it loves lactose. It breaks it down rapidly, very rapidly, and so as a result, you get that metallic green color. Now, these media, okay, they let us study not just the morphology of bacteria, but also the biochemical activity of the bacteria, the behavior of the bacteria. When I say behavior, I don't necessarily mean like, the things the bacteria likes to do, or its personality or things like that. That's a very like animal-centric view of behavior. Behavior is just what an organism does, and how it responds to stimuli to change that behavior. So in the case of bacteria, behavior is very much what the bacteria makes and when and how much and things like that. So that's very much like what we can look at with some of these media as well. Bacteria exhibit a huge amount of diversity and variation in terms of their natural ability, like their biochemical activity, and also their specific behaviors. There are millions and millions of species, most of which we've never described. We haven't described all of them yet. And so we don't even really know the degree of diversity that is out there yet. And we're still learning every day. New species of bacteria are described and discovered every day. And so even slight variation, these slight differences among species and strains can mean a very different behavior. Like, for example, the difference between the bacteria's ability to ferment mannitol or not. The difference in the speed of lactose fermentation, or the lack of that ability. The difference between the ability of a bacteria to resist an antibiotic, or if they're vulnerable to it. And, things like that. Things about, like, can the species produce a toxin or not? Can the species move or not? Can the species, digest this resource and use it as a carbon source or not? Things like that. There's so much. An incredible amount of variation. We, over the course of our class, will use a lot of media to help do what we call biochemical testing. We're going to evaluate these bacteria on their ability to do certain behaviors and have, certain metabolic reactions. Like, do they produce specific enzymes to do specific metabolic processes? This media can really help us evaluate this for these bacteria, and also, in the process, help us distinguish between bacterial species and strains. We use these things as diagnostic tools and methods. And essentially, we can grow bacteria on different media, and based on what they do to that media or what the media does to them, we can use that information to learn more about the bacterium. And in the case of identifying to species, we can use that to help us get closer to its real identification, like what species it really is. Okay, and we're going to be doing this a lot in class, so this is very much something that you will be using throughout the semester. Okay, We're going to be working on this stuff throughout, and getting acquainted with a lot of these tools, and together we'll be working on testing them and using these methods to identify an unknown species.